Hello friends, welcome to Student World. Today we will talk about DNA ligation by linkers. First of all, we should know why we need linkers in molecular cloning. During DNA ligation, DNA ligase catalyzes the joining of compatible ends present on vector and insert DNA. The ligation efficiency depends upon the ends which we are ligating. The efficiency of ligation is high when we ligate sticky ends than blunt ends. So we require linkers to generate sticky ends on blunt-ended DNA. Now, first of all, we should know what are linkers. Linkers are short, usually 8 to 16 nucleotides in length, double standard synthetic, blunt-ended DNA molecules which contain one restriction site within their sequence. For example, EcoR1 linkers contain EcoR1 sequence. Now, uses of linkers. Linkers are used to provide sticky ends to a blunt-ended DNA molecule, which can be vector or insert DNA during gene cloning. The linkers first of all join to one DNA molecule by blunt end ligation, and then sticky ends are generated by cleavage of linker with a suitable enzyme. Now we will see how we use linkers. You see on screen a blunt-ended insert DNA and a linker. First of all, we ligate two molecules of linker to blunt-ended insert DNA with the help of enzyme DNA ligase. In next step, the insert DNA with linkers at both ends is cleaved with EcoR1 restriction enzyme to generate sticky ends. Finally, the insert DNA with sticky ends is ligated on a vector containing sticky ends and a recombinant vector is formed. At last, we discuss the problems we face during the use of linkers. A particular linker is chosen during DNA cloning because restriction site present in the linker is known to be absent from insert DNA. But when we have to clone a gene whose sequence is not known, we cannot use linkers because if restriction site is present in linker, then the gene is cleaved into fragments and we are not able to clone the complete gene. Now, the solution of this problem is either we methylate the restriction site present in the insert DNA so that restriction enzyme is not able to digest it, or we use adapters which have preformed sticky ends. Thank you.
Created using PART-2.